thank you for that lovely introduction. It's great to be here. I'm going to have to ask your indulgence because I thought, because of my position in the program, and also because I was probably the oldest person in the room, I was going to give not a research talk as such, but rather a perspective view of the field of networks, and I hope you will accept that. Certainly we know that networks are incredibly important. They underlie all of the most sophisticated functions of living organisms. James has alluded particularly to networks involving DNA, but I myself am more interested in the networks that occur outside of DNA with protein interactions. But it's clear that they are fundamentally important, and also that in order to understand them, we need to use computers. They're just too complicated to comprehend in other ways. And we also know that when it comes to representing individual proteins, such as enzymes or motor proteins, or individual processes such as action potentials, then computer simulations are able to reproduce data and predict outcomes with as much precision as we like. So this has encouraged people to think, well, all we need to do is just keep doing the same, and we're going to be able to account for everything. And just a couple of expressions of this recently. Many of you may have seen this. This paper was published last year by Carr et al. Entitled "A Wholesale Computational Model Predicts Phenotype from Genotype," in which they took this very simple organism, Mycobacterium, which has only, I think, something like 400 genes, and were able to integrate all of the important functions going on in this cell, all the way from DNA replication, transcription, translation, metabolism, and it was a great technical achievement because they were able to integrate these things, all of these different aspects, so that, for example, the synthesis of proteins was integrated with the function of the proteins in the cell and so forth. So it was a great achievement. And they were at pains to say that this was a first draft and not a complete story. But they do imply, if you read the paper, that they are near the end. So, for example, they say they're predicting phenotype from genotype. And a little way down here they say, if you can't read this, computational model that includes all of its molecular components and their interactions. So it's quite a claim, actually. And I'll raise the question later on whether just how complete such a description can be. And moving to an even more complicated level, we have, of course, the knowledge of the brain, which is of great interest to us and has great resonance with computational investigations. And as you may know, there's various initiatives have been taken to model processes in the brain. The most well-known is Henry Markram in Switzerland, who has, I think I lifted this from the New Scientist. I don't know if you can read it. Neuroscientist Henry Markram says he can build a supercomputer replica of a human brain down to the individual ion channel. Now he has 1.3 billion euros to prove it. And here's a quotation from a conference in Oxford in which he said, "It is not impossible to build a human brain, and we can do it in 10 years. Okay, so these two scientists, and there are many others, Craig Venter and many other people, have this sort of optimistic view that all we need to do is to keep doing what we're doing, use the simulations that we've already developed for metabolic pathways and so forth, and just apply them to more and more complicated systems. But there are many people, for example, to talk about the brain for a moment, I have many colleagues in Cambridge who work on, for example, the development of the retina, where you have multiple different kinds of neurons which emerge during development of the embryo, or people that work on synapses, on the functioning of individual synapses, who find it hard to comprehend how their whole career, the work that they're spending their whole career on, can be so easily sort of assimilated and rendered in a computer program. And so there's certainly another side to this story, biologists in particular, who feel that there are things that are just too complicated at the moment to replicate. And I'm not here, I like to say, I'm not being what Craig Venter refers to as a 21st century vitalist. I'm not saying for a moment there are things in living organisms that are not physical and that cannot in principle be represented. I just question, and this it is a genuine question, whether in fact we have at present the tools adequate to deal with them. So obviously, I don't suppose anyone is able to address all of these issues as directly, but what I can do is to draw my experience of working on what is arguably the simplest form of cell behavior, and that is the behavior that bacteria such as Escherichia coli show when they detect and move towards sources of food and away from repellents. And they do this by modulating the behavior of their flagella. The flagella are driven by motors inserted in the membrane, and the motors are rotary motors, and if all the rotors turn in the same direction, counterclockwise, then the flagella form a nice smooth bundle, and the bacteria swim smoothly in a given direction. But if one or more of the motors reverses transiently, then the flagella break out from the bundle, and you get what's called a tumble, change of direction. And the bacteria proceed in this manner of smooth running and tumbling. And they do this in the absence of any stimulus, but if they sense some nutrient, some amino acid or a sugar, and they happen to be moving in that direction so that the smell, if you like, gets stronger, then they suppress the tumble and they keep going. Conversely, if the taste of food gets less, then they tumble before than they would otherwise. And this is a sort of empirical strategy that works very well. As I mentioned, this is widely regarded as one of the best understood pathways in biology. It's the basic units are shown up here. So you have the 4 or so flagella, each one of which is inserted into the membrane and cell wall in a motor, which rotates at several times, 100 times a second. And then another location on the cell, usually at one end, there's a cluster of transmembrane receptors, which, so to speak, view the environment, the universe, chemical universe surrounding the bacterium, and depending on what they pick up, they relay across the membrane through a conformational change a signal to an underlying lattice of a few proteins, not a huge number, one of which is an enzyme, a kinase, and the kinase adds a phosphate group to a particular protein, Ky, which diffuses throughout the cytoplasm, and the higher the level of phosphorylation of this protein, the more likely the motors are to switch direction to a clockwise rotation and therefore cause a tumble. And that just shows a higher version of the receptor cluster. And this is just, I'm not going to go through this pathway, but just to give you an idea of the main players, each one of these letters represents one of the proteins in the pathway. So here's a receptor, KW is an adapter protein, the function of which has only just last year been really uncovered. The kinase I mentioned is A, which takes ATP, burns it continuously in a futile cycle, and transfers a phosphate group to Y. This is the only freely diffusing protein in the pathway. And YP diffuses through the cell, and depending on its concentration, affects the motor. There's a phosphatase, which sharpens the response, key Z, and then there's another system which is involved in the memory of the bacterium, also seen in adaptation, in which once the receptor is exposed to a certain concentration of attractant, it starts to adapt. And this is achieved by adding methyl groups to the cytoplasmic side of the receptor. And this is, in fact, a form of simple memory that the bacterium has. It doesn't have a spatial sense, it just has a memory, and it remembers how things were a few seconds ago and compares them to the present. Okay, so all of these proteins have been sequenced, isolated, biochemically characterized. This is just a close-up of the receptors showing that they actually form a complex with these two of these proteins. And here's the Y that's diffusing down to the motors. So we have there's a lot of data from genetics, biochemistry, physiology, etc. And it's been a very attractive target for computer simulations. And just to run through a few of the things that we can do now. So, for example, here's a classic response of a bacterium to a pulse of aspartate, which is just added from a pipette, a very brief transient response, which changes. This is the rotation of the motor, which gives an overshoot like this. And here's various. This is in this case a deterministic simulation, which shows the overshoot. Here's a longer-term response in which this is experimental. Bacterium is exposed to a ramp of attractant, and then as the methylation kicks in, it adapts, and here's a simulation, in this case a stochastic simulation. This is an old slide, but just to emphasize the fact that there are a large number of mutants. These are bacteria, after all. It's very easy to knock out or amplify individual components in that pathway, and qualitatively at least, all of these can be predicted in a computer program. The lattice, which is of great interest, lattice of receptors, shown here with fluorescent stain. As I say, last year it was discovered that in fact the receptors, which are grouped in groups of 3, shown here as a little trimers, have an underlying lattice which is made not only of the kinase, the key A, but also this mysterious protein key W. And it turns out that the A and the W together form a hexagonal lattice, which at least we propose, and this is still under investigation, is a substrate for a spread of conformational changes which lead to the amplification of a signal. And I'll just give you just to give you a flavor for the sort of simulations we run now. So each one of these, each one of these trimers is a trimer of receptors, which in this case we're looking at from the outside of the cell looking in. And if we look on the other side, from the inside out, we see that here's this lattice of key A and W, and active, which is either active, which is shown in orange, or inactive, which is shown in gray. And because of what we propose is this conformational spread, the activity propagates in clusters over the array, and the effects of this are seen when we add a stimulus. Sorry. So if we add a stimulus, we get a very rapid response, which then slowly adapts. So we can reproduce the broad features, certainly, of the response in this manner. and we can get suitable amplified responses to not just a single stimulus, aspartate, but the system will also respond to aspartate and then respond separately to serine, and these give different methylation patterns as expected. And just finally, we've also built models of the motor, stochastic models of the motor as it switches from clockwise to counterclockwise. And on the left, there's a series of experiments made by, in Oxford by Richard Berry and colleagues, showing an individual motor that's switching from counterclockwise to clockwise. You see how it's highly noisy and how there are pauses and reversals and so forth. And this is a model that they used, which is based on our model, to simulate the same behavior. So, you know, we're getting pretty close. It's true that it must be true that we have a broad understanding of this system. Now, do we understand everything? And the answer is actually no. Let's go back to this little pathway. First of all, I've only been talking, or mainly about one receptor, one of the five that are there, the aspartate receptor, and certainly most experiments are performed with either TAR or TSR. Very few experiments are carried out with the others. So the bacterium actually responds to something like 50 different chemicals. So a lot of those have never been even looked at experimentally, let alone modeled. The chemotaxis pathway doesn't exist in isolation. It's in the cell together with everything else, and as you might expect, it's influenced by everything else. And one of the prime influences is actually another pathway which takes up glucose, the HPR system, shown here, but there's two proteins which actually feed into the Ki. So they change the swimming of the bacterium by affecting Ki. So a complete picture would certainly have to include those. It's been known for many years that this little protein Ky that diffuses through the cell is not only phosphorylated, but has sites of acetylation, I think 3 or 4 sites, but for a long time it's not been understood what the function of this is. It now turns out that, first of all, that acetylation is ubiquitous in eukaryotic as well as prokaryotic cells, as you know, and probably in most situations reflects metabolic activity. And indeed, what it seems that the acetylation of Ky is doing is when the cell is replete, has plenty of food, then it actually inhibits the action of Y on the motor. So, in fact, the cell stops swimming so actively, and it's now poised to actually enter a different phase, which is the formation of biofilms. Then, a few years ago, there was a very dedicated high-throughput screen of mutants which affects swimming and chemotaxis that was performed. And this uncovered at least half a dozen genes which had not been previously identified, and a number of these affect, in fact, the motor and the control of the motor, and some of these were involved in a cyclic GMP pathway that again is involved in the switch to biofilm formation. And I have to say that this screen, although it was pretty thorough, was still pretty coarse, and there are many subtle mutants that would never have been picked up in this way. Another thing that's come out just in the last couple of years is the motor itself, which we think of, which we had thought of as just like a human-made machine, because it's got a number of components, it rotates very efficiently, as being, as it were, made of metal and immutable. But that's not the case at all. And it turns out that a number of the components, so here's the flagellum which goes off out of the ceiling. This is this whole apparatus here rotates, and then there around this is this cylindrical structure. There is a number of static units which, among other things, funnel the protons across the membrane. It turns out that a number of these proteins are actually rapidly exchanged in the cell, and in fact their numbers can be altered depending on the experience of the cell, and this comes from last year a paper from Howard Berg's lab in which they took a bacterium with a truncated flagellum here, and what they did was simply move the bacterium up until it contacted a bead that was held in laser tweezers, and they observed the rotation of the motor. And the motor originally, before it made contact with the bead, was rotating around 300 times a second. The minute it contacted, fastened onto the flagellum, the speed fell precipitously, a few tens of rotations per second, because the load was much increased. That they then observed a progressive, stepwise increase of the rotation up to a new plateau. And it turns out that this was due to the recruitment of a protein called MOP-B to the motor. And with very light load, there were only one or two components in the motor, but as the load grew bigger, it went up to something like 11. So here's an example in which the motor, which is quite unlike a human motor, it's ultimately very flexible and can change its function with the surroundings. How am I doing for time? Lastly, and in my mind, the most interesting aspect is that of post-translational modification, and that's illustrated by the cluster of receptors that I've already referred to. So these are at one end of the cell typically, and there's something like 3,000 or 4,000 of them, and they're grouped in threes, as I mentioned, and the four different kinds are distributed among these in this lattice. And the methylations that I mentioned, which serve as a memory mechanism, are shown as little dots here. So each receptor is itself a dimer, and there's a trimer of dimers. Each dimer, each individual protein has, in this species, 8 sites of methylation. So you can have everything from 0 to 8. And of course, you have the potential of having those methylations in different positions. So the amount of combinatorial combinations is astronomical or not possible to count. So this is one of the receptors. There's the structure, and this is its sort of diagram of it in the membrane. Here's the membrane. Here's the external part that detects the molecules of interest. Here's the cytoplasmic part. And this is the part that interacts here down at the bottom with the kinase. And here are the sites of methylation. And now when we write models for bacterial chemotaxis, depending on the level of sophistication of the program, we use different representations of the methylation. So the simplest of all, which was an old program that we wrote many years ago called BCT, simply represented the level of methylation as a single variable. So methylation varied from 0 to 100%. That's it. A more sophisticated model, which Carl Firth in my lab developed, which was a stochastic model, and took into consideration the free energy changes of the receptors as they flip from one state to another, now had identified 4 sites of methylation. And each one of those could be assigned a different occupancy, and each set of occupancies would specify a particular free energy. The reality, of course, if we want to include all of the combinations, is that there would be at least 256 of these, each one of which is potentially a distinct structure. Now, I'm not claiming that each structure does a different thing. Of course it's not. Most of this is noise. But what I am saying is that we don't actually know how much of this potential information is used. And just a nice illustration of this fact comes from a study from Lipchaber's laboratory a few years ago, in which they looked at the thermal response of bacteria. Now, bacteria, E. coli, swims to an optimal temperature of around 30 degrees. And in fact, it's the same receptors that I've been talking about, the ones that detect sugars and amino acids, which are involved in temperature sensing. So there's one receptor actually avoids excessive heat and will give a tumble if it gets too hot, and another kind of receptor avoids excessive cold and will give a tumble if it gets too cold. And the balance of these two ensures that the bacterium moves to the optimal position. So what Lipchab and colleagues did was to take a culture of bacteria and shine a little spot from a laser to warm up one small spot in the culture medium. And what they observed was that if the bacteria were from a rapidly growing culture, then they would congregate in this warm spot, and that's shown by the red cells there. Those were taken from a rapidly growing culture. But what they also found was that if they took a culture that had been grown to stationary phase, where the medium is exhausted, same bacteria, presumably same genetics, and did the same experiment, then they avoided the hot region. Now, what sense does this make? Well, from a teleological standpoint, you can argue that when the cells have plenty of food, it's in their interest to grow as fast as possible. They want to go to the warmest or most appropriate temperature for division. But when resources are limiting, that's not what they want to do. Under these conditions, they want to, so to speak, chill out, slow down, and perhaps seek some alternative way of surviving the dearth of food. And it's known that what happens in these transitions, this simply shows a way of measuring the change of thermotaxis with the culture density. It's a very sharp transition here. And one of the changes, longer-term changes, is that there is a change in the numbers of the two different kinds of receptors. So when you're in a very high-density culture, your cold-seeking receptor becomes less effective, and it becomes less effective partly because it becomes methylated through the secretion of some molecule from the cells, but also eventually there's a change in the composition of the receptors themselves. So this is something which would not be in a model unless you included it specifically. So sort of standing back at this, and as you know, this is just the tip of the iceberg. This is a very simple system I'm talking about. This pattern of methylation of the receptors acts in something rather, and it's been likened to a buffer between the genotype and the phenotype. So the genotype does not specify the phenotype, no matter what anybody tells you. What the genotype specifies is the composition of things inside the cell, which then interacts with the environment, and depending on what the environment tells it, that generates the phenotype. And that's just shown schematically here. Okay, so this is bacteria. So what we do not know about E. coli chemotaxis, there's still many genes being found, and almost certainly there's a lot which haven't been found because the screens aren't sufficiently sensitive. All of the genes in chemotaxis are controlled in some fashion, and we really have very little idea at the moment about what controls them and how they're controlled. Most proteins are modified by acetylation, phosphorylation, methylation, and these modifications don't exist in isolation, but each one affects neighboring proteins, and there's cooperative effects, which again we don't understand. And there's a question of when a protein is made in the cell, what it interacts with. And there's no way that I know of being sure that, shall we say, any two proteins in the cell can never interact. How could you ever prove it? If I said that protein A and protein B in a cell never interact, how could you prove it? I could always say, well, you could say, well, I do all these tests and they don't interact. Well, I said, well, you took it in, you didn't methylate this protein. or you didn't express it in the right context with the right complements of small molecules, and so on. So the potential is for any two proteins in a cell to interact, even at some level. Okay, so at this point you're probably saying, well, this is splitting hairs. We understand how chemotaxis works. Why are you bothering with all these details? We're not interested in how a bacterium responds to some strange sugar or the last details of what goes on in the receptor cluster. A protein, a cell that, for example, lacks, shall we say, the ability to modify its motors in the manner that I described, would still be able to grow. It would be in a laboratory, you wouldn't be able to see anything wrong with it. All that would be wrong would be that when the new flagellum was made, instead of turning it slowly as it should, it would spin like crazy, and so it would burn energy. So that particular mutant would not be able to compete with a wild type that could adjust its energy budget much more closely. So these things really would create what you might call a sick cell. But it would work. Now it comes to the difficult part, which I mean is a genuine question. I have the feeling that these things which are sort of fine-tuning for E. coli assume more and more importance as we go up the phylogenetic scale. And when we get to things like development of animals, all of the things that I've been talking about, post-translational modifications, control of gene expression, and many others, are magnified in number, and I would suspect of importance. And I'm not sure that we are really able to cope with that. Now let's jump all the way from the bacterium to the most complicated system that we know of, the human brain. Here's a beautiful picture from Ramona Cajal showing a portion of the cortex, and these are pyramidal cells, as you know. These are nerve cells, and these are their dendrites and axons. And you know that these carry electrical signals which underlie all of our mentation. And each individual pyramidal cell has on its dendrites and axons, dendrites particularly, numerous points of contact with other nerve cells, which are synapses, and each synapse is not unlike the bacterial receptor plaque in size, and mediates the connection between those two adjacent cells. And what the brain does depends on which synapses are there and how they operate. Now, how far are we along in knowing how this works? Well, as I said, the whole system is replete with complications. First of all, which nerve cells are there and what kinds of nerve cells, and their shapes depend on a very elaborate program of gene expression. And this is taken from a much simpler system, and it's kind of old now, but this is just one portion of the wiring, if you like, the gene expression network of a sea urchin embryo. And each one of these is a little stripe thing, is a gene, and just gives you an impression of the complexity that occurs in this developing system. And we can only imagine how much more complicated the brain it would be. Something that doesn't occur in bacteria is RNA splicing. So each gene can itself be, as you know, cut up into pieces and recombined to make a large number of products, sometimes a ridiculous number of products. So, for example, this protein in Drosophila, I've forgotten what the number is, but it's many hundreds of variants are formed. And in the human brain, there's proteins such as neurexin, which are similarly diverse. And the question, is, of course, of which spliced variant forms and when and under what conditions is still a largely unopened book. Protein modifications in eukaryotes is a huge subject. And there's a large number of different kinds of modifications. Phosphorylation is still the most abundant, but all of these other different forms of modification. Most proteins, is my impression, are modified in at least one, have at least one modification. Many have numerous different kinds of modifications. And exactly the same criticism applies that I applied in the case of the bacteria, and that is to say that the level of the kind of modification that occurs is going to depend on the environment. It's also going to depend on which protein is next to which protein, and the combinations of proteins will have effects which they don't individually. So this is, to me, a sort of an unopened box that we really don't know how to tackle at the moment. So, and finally, of course, the synapse itself. Here's one synapse, and is a mitochondrion. So what I was talking about before is the bacterium is about that size. So it gives you a sort of flavor for the complexity that's going on in this synaptic ending. It's almost certainly going to be plastic, just as the flagellar motor is, when we know that already that there are components which come and go and which are amplified and attenuated depending on conditions. So I'm really in two minds at this point. When I think about all of these complexities, I wonder whether, in fact, if we can continue, whether we're going to have a satisfactory description of them using existing methods. So what's special about the networks, particularly the protein networks themselves? First of all, you can have connections over long distances because of the free diffusion of components. So in principle, any component can influence any other component. That's not true of a microchip. That's not true of a silicon device. So the elements are highly flexible and capable of an essentially infinite modification in both activity and connections. This is very different to the way we normally think of silicon devices. Input and output devices are built from the same structures, and this comes up in the case of the modification of the flagellar motor. For example, if we think of a muscle, human muscle, which is built of a highly regular arrangement of proteins, we know that this is incredibly plastic, and depending on exercise, nutrition, age, all these factors, it will be a different structure. It's not as though it's one mechanical structure that's permanent. And this is because the very same components which are used in signaling are actually used to build the structure as well. And again, that's a twist which we don't see in silicon devices. And of course, probably most important of all, the pathways, the networks that we see have been part of evolution, and they've been continually updated and optimized for performance. So I'll leave it there. I don't know how far we can get with the sort of existing approaches, and whether there is any possibility of perhaps adopting new approaches, possibly even new hardware, in which we develop systems which are resemble more closely these features of the living world. Thank you.